Hello, everyone. Welcome to my 3-minute thesis on H3K4 methylation and myogenesis and its dysfunction and disease. Now, what is myogenesis? Myogenesis is the creation of muscles from stem cells, going from satellite cells, which are a type of muscle progenitor cell, to proliferation of myoblasts, which are basically just a lot of satellite cells, into combining all together into a myotube, which is a multinucleated or multinucleus kind of cell. And then all of those combining together to become muscle fibers as we know it now. Now, myogenesis is obviously important because without muscles, we wouldn't be able to live. But what is H3K4? H3K4 is an epigenetic term. Now, what is epigenetics? Well, imagine your genome is a painting canvas. You can't really change it without damaging it, but you can change how it looks. Epigenetics is the paint. Epigenetics is the study of changes in gene expression without changing the genome itself. Now, this can happen through histone modifications. Histones are these proteins that your DNA wraps around in order for either it to be silenced or activated. Now, H3K4 is one of the activators where the DNA starts to unwrap from the histones so it can be readable by RNA polymerase. This creates mRNA as we know it, which is gene expression. Now we have histone modifications such as H3K9 methylation, which leads to repression because the DNA wraps itself around the histone tighter due to charge differences. Now, what I'm studying is specifically the third histone and the fourth amino acid, which is a lysine. Now, as I diagram down below, methylation can be added either in 1, 2, or 3. The changes in how much methylation occurs changes also how much gene expression occurs, which 1 being the least or 3 being the most. But with repression, 1 being the least and 3 being the most repressed. Now, why is H3K4 important? H3K4 is important because it is an activator of specific and non-specific genes. We need H3K4 to be methylated by other things such as MLL1 or 2 in order for myogenesis to occur because we need the correct timing of these genes to activate. Now, H3K4 helps us by activating these genes, and then we have demethylators such as LSD1 that take the methyl groups off, have H3K4 be unactive, and then have another group be active instead, so then the genes can be repressed. So H3K4 is important, but if H3K4 is not properly regulated, this can lead to diseases because certain genes will be turned off when they need to be turned on. This can lead to Duchenne's or fasciohumeral muscular dystrophy, but either way, H3K4, pretty important.